Hi, Mark. Just to let you know, we're now live. So, welcome and thanks for joining us. So, the second session will actually feature our event partner, Strateos. We just heard from Peter on the last session. Now we're going to hear from their CEO, Mark-Colbrie, and he will share his thoughts on paving the way for the next generation experimentation with self-driving labs. So, I will turn over to Mark as the presenter for the session. Over to you, Mark.
Great. Thanks to you so much. I'm so appreciative of the opportunity to talk about, which I think is the wave of the future in terms of a new era of discovery capabilities within life sciences. And within that context, there's a lot of wood to chop to get to the ultimate goal of having self-driving labs and artificial intelligence assisting in discovery. And the first and foremost is there has to be a radical improvement in reproducibility. And whether it's this Nature article or published data from Amgen and Bayer, it's quite evident that there are tremendous problems with respect to replication of data. And our view is, in order to alleviate that initial problem, is that fleets of automation need to replace manual workflows. And the old style 1850 artisan type of work needs to be brought forward into full modernization with the ability to generate rapid data with high reproducibility and high fidelity. In turn, those data set streams and sets can drive machine learning models to do the next leverage effect on those data streams. And what that means then is there's the need for creating unification of lab experiments that are run internally and the ability to tap into sophisticated automation modules over the cloud with a hybrid cloud lab software control, as well as to integrate a variety of machine learning tools to drive discovery more quickly. So within that context, if you were to look at what is a self-driving lab and how that might operate, it's really the ability for unified design, make, test, analyze cycle for creation of new molecules, generation of new data, with those data streams being fed into machine learning models that can be done on a continuous improvement basis for rapid iteration of experiment. So one can think of this as a very large instrument rather than the individual components, in the context that you want to have the ability to put experimental data controls in and get modifications done to your machine learning models to advance and accelerate discovery processes. So within that context, the Strateos Smart Lab platform is really pushing towards turning life sciences into an information science. And the ability to generate clean data sets with fair data standards, which, as you know, encompasses findability, accessibility, interoperability, and reusability, are pretty essential for accomplishing that goal. And it's interesting to me in talking to folks outside of life sciences, they sort of assume all that's happening today. And the fact that it's not is a direct requirement of being able to come up with new software and new ways of integration of automation with data science tools to really advance science. And so in order to accommodate that, you can really think of labs as data centers. And one wants to be able to control the experiments that you're running, to be able to do sophisticated scheduling of those experiments on a fleet of automation platform, the ability to have consistent protocol designs that are easily shared and communicated. There are important elements of not just the raw data of what is happening with respect to the machine output, but also the metadata of environment and machine condition that can go along perpetually with the hard data coming off the instrumentation and experimental runs. So that means that there's a lot of software that needs to be developed and incorporated in order just to make those data run and integrate that with a unique automation and robotic platform In our context, that's something that we don't see anywhere else in the world today, with the ability to do both chemical synthesis and biological testing and drug discovery, as well as apply our approach across a wide variety of synthetic biology areas. What that means is then there's an entire platform here of integration of software, robotics and automation, cloud access, and artificial intelligence tools that encompass our entire platform. And so that means that Strateos has the ability to control workflows within our own laboratories and our own automation streams, as well as have created a software architecture that can be deployed in existing labs and be able to toggle back and forth between those two locations with common protocols and common experiments to do the design, make, test, analyze a cycle irrespective of geography. So that software activity then is one where it's pretty sophisticated because it encompasses, in addition to the operational aspects of the experiments, intelligent protocol design, smart workflow management, very sophisticated experimental scheduling, the ability to control things down to the device level, as well as an automation module level, as well as stitching together automation modules across a variety of modules to be able to run things all together. And that also encompasses the ability then to obtain data and push data out across that entire platform. And within the context of Strateos, that's a situation where we have two primary labs, one in San Diego with over 20 plus automation modules that is more focused on drug discovery, and one in Menlo Park, California with 5 automation modules that enables a whole slew of synthetic biology applications. And really what we see are the opportunities for entire workflows of life sciences across the board, irrespective of whether it's small molecule, large molecule, protein synthesis, synthetic biology, CRISPR work. All these things are amenable to this type of platform. And here you see just a portion of our automation modules in San Diego, California, and the ones in Menlo Park. Included in this image are the important features of transparency and traceability of what's happening at workflows and where things are occurring during the processes. And really what this means is the tremendous ability to have access to automation that people would not normally have even a remote chance of being able to tap into. And those include the ability to maintain and store samples of different types, the ability to do medium to high throughput screens, the ability to have automated chemical synthesis, to create a large instrument that combines chemical synthesis with biological testing in a novel capability, so that all these workflows then are co-located to really work on hit expansion and lead optimization activities across the board to accelerate early stage drug discovery. And really, if you look at what's gone on historically, this is a phenomenon where chemistry has been often highly manual. There's a lot of work that has to go in. It's very artisanal related to that work. And this is then one where it's supplemented with contract research organizations to try to get work done in addition to work in internal labs. And we see the world as going forward to an integrated laboratory of automation combined with artificial intelligence capabilities to change the concept here so that you can have very effective design of experiment to iterate quickly with high fidelity data to drive hypotheses forward. And I'll go through this with specific examples. We're doing this in the synthetic biology world under a DARPA project to optimize the creation of new biosensors. And that's one that requires multiple groups and multiple coordination across a variety of organizations to achieve the goal of machine learning driven hypotheses to accomplish this task. There are tremendous challenges in doing that in terms of just even making sure experimental designs are proper, that data coming back are appropriate, and just even curating all those data streams is difficult typically to do that. But we're able to accomplish that. And the direct result of this is the ability to accelerate communications and activities that would take weeks to take down to days, as well as eliminating errors in communications, as you can imagine, trying to coordinate all those groups. And what that means is we also have the ability to generate new data streams that can feed into and modify the machine learning models that have been generated in order to achieve this optimization of biosensors. And in turn, what that means is that you can generate new hypotheses, push those new hypotheses onto our platform, have the data push back into the machine learning models, and have an iterative cycle of continuing to advance this. And in this case, this is the ability to create new biosensors, but obviously this has a direct impact on basically any synthetic biology application. We see a parallel work going on in Dr. Romero's lab in Wisconsin with the ability to use artificial intelligence algorithms designed by that outstanding team there, and to tap into a lab automation platform to accelerate that design, make, test, analyze cycle for synthesizing proteins. The challenges for that historically for Wisconsin would have been slower cycle times, the lack of data that could be done real time, and the difficulties of doing things at scale to accelerate and confirm the AI models. And by being able to tap into the Strateos platform, that's one where integration of automation and having automated protocols means greater ability for throughput, tremendous reduction in cycle time that accelerates discovery, and the ability to tap in and use these activities with basically zero hands-on time by the folks that otherwise would have to be standing under hoods and pipetting operations. The same kind of approach that are used in synthetic biology and in protein synthesis can be applied to new drug discovery. This is a pending project that we have with using in silico designed compounds to do the same kind of work, and in this context, specifically targeting antiviral molecules for a variety of different indications in the world that requires new antivirals to be developed. So we're excited about this type of a program and integration, and we're kicking off additional projects in this drug discovery area with this type of approach. The challenges there are, again, typically slow cycle times, whether compounds are being synthesized internally or whether they're being through an outside party. The types of workflows that people need to do are distributed often in different cities that in turn requires additional cycle time. And the ability to tap into our platform means the opportunity for combining automated chemical synthesis with biological testing to generate promising leads that have been initiated with the activity of using virtual screening for the creation of the initial set of proposed compounds. So we're very excited about what we think is going to be a tremendous breakthrough related to drug design and testing and accelerating those early stage cycles. So really, this is a situation where Strateos is seeking to be a major fulcrum for moving laboratory science to become an information science. And so we're very excited about this type of capacity and capability that really brings life sciences forward into complete modernization with a flexible, automation platform that people can control, they can program, they can have direct access to data and a full transparency of data streams and where things are in their workflows, and the ability to take those clean data sets to use a variety of data science tools to further leverage these activities. And again, ultimately, with human intervention can be accommodated every step along the way, but the ability to use machine learning as an assist to drive this lab experimentation is now a real possibility. So we're super excited about this platform. And with that, I will be happy to open up this, for questions. So thank you for your time.
Fantastic. Thank you very much, Mark. Great presentation. Can you hear me okay?
Yes.
Perfect. So yeah, great session. I think we'll push it back out to the audience again. So the last discussion was quite lively. There was a lot of questions being asked. This is your opportunity to ask some direct questions. So I've seen the first one come in. But just to remind everyone, if you go to the right-hand side of the screen, there's several tabs. Click on the one that says Q&A and drop your question in there. The first question, would you like me to read it out for you, Mark?
Well, I can read it. The first question is, how many customers do you have using our platform? We're very pleased that we've got a tremendous variety of customers, everybody from academic institutions to virtual companies coming out of incubators to small, medium-sized biotech and to large pharma. So we have a very broad distribution of customer type, and we have a very broad distribution with the types of workflows in the context that this accommodates drug discovery, synthetic biology, and ultimately, as we continue to add to the platform, we'll continue to expand the capabilities of that environment. We have tremendous experience at cloud interface for how to manage all those kinds of activities that are incorporated into our software that enable very sophisticated scheduling across the entire platform with full information security, so there's no chance of any kind of information being inadvertently shared with other folks. And this is a situation that visibility of activity is very, very important. And that leads to the next question. Is there an over-the-shoulder camera in case something goes wrong with experiments, especially with cell-based assay? The answer is yes. People can literally watch experiments if they want to. They can also obviously get notifications if they want to. But more importantly, this is a situation where you can watch on the scheduler also what's the next step, what's going to happen, what are the expected time periods for that kind of work. And what's nice about that is the ability then to control your experiment with computer code. You can actually program the lab yourself to do the work that you want to have done. And that's an incredible ability to reach in and under a flexible automation module, be able to control it yourself if you choose to do that. Or you can pre-pick a series of other experiments where you can vary the parameters if you were to choose that as well. So we're basically excited about that. So let me jump to another question. Fantastic talk. Are there also economical arguments to run experiments? Yeah, no question about that. This is a situation of, first of all, time is an incredibly important factor. We know that we've run certain experiments where we've designed compounds and run results, and we know those can be done ridiculously fast. In fact, in some cases, the more extreme cases, 48 hours from design of a compound to synthesis to biological testing. But inherently, we're in a situation where we can do things in days that would normally take weeks. So time is a huge cost factor for getting drugs to market. But more importantly, because these workflows are co-located, you're in a situation where you'd reduce a tremendous amount of overhead related to running experiments. So we're able to have very low cost per data point around those activities. So let's say next question is, how do you deal with the politics of applying machine learning that by definition will change the skill set required of the lab? Hey, we're pretty excited about the concept of assist. And that's the ability to have people who are trained in, say, immunology, focusing on their training. And this is no different than any one could have asked that question related to, oh my gosh, if I do automated, pipetting with acoustic dispensing, gonna, affect my job. The reality is, we see the incredible opportunity for people to be extracted from mundane operations that cause tremendous headaches for people that they were never even trained for those kinds of activities. So we're very excited then about the ability for data science capabilities tapping into clean data sets to be able to allow people to think about, well, what is that new hypothesis? And here's a suggested new hypothesis. Here's some thoughts coming off of the data streams coming forward. So my sense is that the skill sets inherently then will actually line up even better with people's training, and it's because they're not burdened by these tremendous taxes of overhead costs of just trying to make equipment run or work through third parties on a very standalone basis to be able to generate data. Are you focusing your efforts more on providing robotic cloud labs or deploying, our software that we call the Strateos Common Lab environment onto the customer site? We have the opportunity and the ability to do both. That's a phenomenon where we think it's going to be a terrific tool for people with their own existing laboratories that would have the ability to tap into our cloud lab with a common platform of the hybrid cloud lab. You could toggle back and forth between location on demand, but more importantly, even the internal operations can be improved with our command and control software structure that we're releasing related to improving operational efficiencies. This is another gap today, surprisingly, related to just the ability to do broad scale control, scheduling, traceability, and metadata tracking across the platform. So inherently, we are a software company aligned with a tremendous scientific talent, with tremendous automation skills. So we bring all those things together, and it's our software that is being used for both the cloud lab activity as well as the ability to deploy software elsewhere. Is there a specific workflow? What's nice about our platform is there's a whole series of specific workflows that people can do. In the context that we move plates and vials around, we've got a tremendous variety of automated liquid handlers. We've got tremendous opportunities for different readers and different capabilities. So there's a wide variety of workflows on drug discovery, biochemical cell-based assays for being able to generate data. There's a wide variety on synthetic biology applications as well. So we're able to accommodate quite a few things, and we really see the opportunity for acceleration of lead optimization and thinking about these things as design of experiment, and to think of our platform not as a fractionated partition set of individual steps, but think of it as one large instrument. And wouldn't you love to have an instrument that you could rapidly change your experimental design to test hypotheses quickly and get clean data sets back is really the opportunity. There's a follow-up question on deployment of a customer site. How generalizable is the solution? This is one where our software capabilities are extremely generalizable, and this is one where we've learned a tremendous amount for deploying software, not just in our own labs, but also on behalf of Eli Lilly and Ginkgo Bioworks. And not only do we have direct ability to talk to instruments, but we have generalizable capabilities to do command and control software across internal labs that range from small labs that can have very simplistic but essential software for being able to have visibility of what's happening in one's own lab to very, very large labs across the board. So there's tremendous flexibility in terms of the types of labs that can utilize our software. And it depends also where they are in their life cycle, where they might want the initial offering that we call Collaborate, which provides great visibility into their own operations, all the way through to our large capabilities that control and manage fleets of automation, as evidenced by our San Diego operations.
Perfect.
Yep.
Okay.
Yep.
So I think that brings the session to a close. We have 2 minutes, and one more final question has come in, Mark.
Yeah,
you happy to take that question?
Yeah, another question. Yeah, how is IP dealt with? Customers have no issues. It's their own data. It's their own IP. It's all controlled by them. They're the ones who have, access to that. There's no risk of, cross-pollination. And so the architecture accommodates where people control their own IP. We've passed information security audits around this too as well. And as you can imagine, we're working with large pharma, and large pharma obviously would have tremendous needs related to information security, and we're well covered there.
Perfect. Thank you very much, Mark. Appreciate the insights and sharing your time with the audience. Great to see that the audience are asking lots of questions and very interactive. So hopefully that will continue throughout the day. What I will say is Strateos have a virtual booth. I may have mentioned this before, but you can meet with Mark and the team. You can meet with them in their booth. You can ask direct questions. You can have face-to-face conversation with them. You can schedule a meeting. You can go in and you can pick up some materials. It's very simple to access that. All you have to do is go to the left-hand side of your screen. There's a button that says Exhibition. Go in there and just enter their booth, and it's self-explanatory from there. So would encourage you to go meet with them. Seems like there's lots of questions. I'm sure there's plenty more that people want to ask Mark and the team directly. So once again, thank you very much, Mark. Appreciate your time on that. So before we move into the next into the networking session, we do have something slightly different. We have two of our other sponsors. They'll be taking you through a technology demonstration, which will focus on their unique session. So the session lasts 10 minutes, and you'll need to select which session that you wish to join. So we have two sessions running at the same time, one with Smart Labs and the other with TetraScience. So just to let you know, after that, we'll break immediately after the session. And that is when, that's a session that's designed for networking. So again, that's where I'd encourage you to go see Mark and the team and the other sponsors as well. Other than that, thank you very much once again. And that concludes that session. See you shortly for the next session.
Thank you so much.
Take care.